Welcome to your daily dose of science news. Ready to dive into some really cool breakthroughs. We've got computational biology, RNA sequencing, even a peek into the future of DNA storage.
It's amazing. These fields are moving so fast, It seems like every day there's something new.
Let's jump right in with drug discovery. Researchers at Mount Sinai have developed a computational tool. They're calling it iDOMO.
iDOMO.
And it's designed to predict drug synergy. Find those combinations of drugs that work better together.
Oh, yeah, like instead of just one drug, you use a few and get a much bigger impact.
Exactly. This could be huge for personalized medicine, right?
Definitely. Imagine doctors using iDOMO to design personalized treatments for each patient based on their genes and everything.
Tailoring treatments. I love it. But timing matters too. It's not just about which drugs, right?
Right.
A study in genomics, bioinformatics, and bioinformatics looked at monoclonal antibody treatments for cancer. And timing was key.
Absolutely crucial. This study showed that delaying treatment until day 8 actually made the antibodies less effective, even when they were combined with other therapies.
Wow, so starting treatment early could make a huge difference.
Early intervention is so important. It seems like the tumor microenvironment changes over time, and that affects how well these treatments work.
So there's a window, almost, where treatment is most effective. Fascinating. Okay, let's move on to RNA sequencing, another field that's just exploding with new insights. There's this new tool, PbImpute. It's really boosting the accuracy of single-cell RNA sequencing analysis.
It's a game changer, honestly. You see, single-cell analysis looks at gene expression in individual cells. PbImpute filters out the noise. It tells us which genes are truly off and which ones just look that way because of technical glitches.
So we get a much clearer picture of what's happening inside each cell?
Exactly. That kind of precision is crucial if we want to understand complex biological processes.
And speaking of exciting discoveries, RNA sequencing is also uncovering some really cool targets for cancer immunotherapy.
Right. So you know how a single gene can create different versions of a protein? It's called alternative splicing.
It's mind-blowing how much complexity there is in our genes.
It is. And it turns out this splicing can create unique tumor-specific antigens.
Antigens. So these could be used to trigger the immune system to attack cancer cells.
Yes. Imagine personalized cancer vaccines or other immunotherapy approaches tailored to those specific antigens.
That's incredible. It could open up new avenues for treating really challenging tumors.
Like glioma. It's definitely a field to watch.
Now, get this. Parse Biosciences just released a kit that can sequence 5 million cells at the same time.
5 million. The scale of this is just amazing. Think of it. We can now study huge cell populations, identify rare cells. This could be huge for drug screening, early disease detection, so many things.
It's like having a superpowered microscope for cells. It's mind-blowing. Okay, switching gears a bit. Let's talk about DNA storage and a potential game changer in the sequencing world. Roche just unveiled their new SBX sequencing technology.
I've heard about this. It sounds pretty amazing.
What's really unique is that it can attach large molecules to nucleotides, the building blocks of DNA. And it still allows polymerase, the enzyme that copies DNA, to do its job.
Wait, so it's like attaching extra information to the DNA itself.
Exactly. Think about it. We could sequence proteins or other molecules along with the DNA. We could attach fluorescent tags to visualize DNA's 3D structure. The possibilities are endless.
It's like turning DNA into a multi-tool for research.
Some people are calling it the iPhone of sequencing. It could be that revolutionary.
If Roche delivers on its promises, accuracy, speed, affordability, it could really shake things up in the sequencing market.
Definitely. And speaking of DNA's amazing potential, there was this really cool study in nature.com about a new approach to DNA-based data storage. It uses coding theory and deep learning.
Oh, yeah, I read that one. It's addressing some of the big challenges with DNA data storage, like making it scalable and robust.
I mean, the idea of using DNA to store data is amazing, right? It's so dense, you can store a ton of information in a tiny space. And it lasts forever.
It's nature's hard drive, and it's way more efficient than anything we've come up with.
So how do coding theory and deep learning fit into all of this?
Well, coding theory makes sure the data is encoded and decoded accurately, even if there are errors during storage or retrieval, and deep learning optimizes those processes, makes them faster and even more reliable.
So we're building super smart algorithms to read and write data stored in DNA. Amazing.
This research is bringing us closer to a future where DNA data storage is a reality. It's not science fiction anymore.
It's mind-blowing to think that one day we might store all our data, our photos, our videos, everything, in tiny strands of DNA.
It's a testament to the power of interdisciplinary research, bringing together computer science, engineering, and biology to create truly innovative solutions. Welcome back. We've got more exciting science news to explore.
Let's talk about something that might seem kind of technical, but it's really important for research. Batch effects.
Yeah, batch effects, they're often overlooked, but super important, especially with these massive data sets we're working with these days.
Right? It's all about data quality, making sure our findings are reliable. There's a great article in Genomics that really highlights this.
Yeah, I saw that one.
So for those who aren't familiar, could you explain what batch effects are and why they matter so much?
Sure. Imagine baking 2 batches of cookies, same recipe, but you use 2 different ovens.
Okay, I'm following.
Even with identical ingredients, the cookies might turn out a bit different, right? Because of variations in the ovens.
So it's like those unintended variations that creep into experiments.
Exactly. Batch effects are variations that come from differences in how samples are prepared, the reagents, the equipment. Even the day the experiment is run can make a difference.
Wow, so many factors.
And these variations can really mess with your data. You might think you're seeing a real biological effect, but it's just an artifact of the batch effect.
That's a bit scary, actually.
It is. That's why scientists use all sorts of statistical methods and computational tools to identify and correct for these variations.
It's like adjusting the oven temperature to make sure your cookies bake evenly.
Exactly. It's a crucial part of ensuring scientific rigor.
Okay, let's move on to something a bit more exciting. The search for a universal flu vaccine. A study in genomics, proteomics, and bioinformatics caught my eye. It looks at a new approach that targets a specific protein on the flu virus.
Oh, yeah, this is interesting.
You know how the flu shot is a bit of a gamble each year? A universal flu vaccine would be amazing.
It would be a game changer. So this study focuses on a protein called CD20. It's found on B cells.
B cells, those are immune cells, right?
Yes, and the researchers are exploring if depleting B cells could help reduce how severe the flu infection gets.
Wait, so you're saying we reduce immune cells to fight the flu? That seems a bit counterintuitive.
It might sound strange, but sometimes an overactive immune response can actually cause more harm than good, especially with the flu. All that inflammation can be damaging. So this research is about fine-tuning the immune response.
Finding that balance, making sure the immune system fights the virus effectively, but doesn't go overboard.
Exactly, and the study showed some really promising results. They found that depleting B cells with a monoclonal antibody treatment actually reduced the development of atherosclerosis in mice infected with the flu.
Hold on, atherosclerosis, that's the buildup of plaque in arteries, right? I thought we were talking about the flu.
We are, but it turns out the flu can actually worsen atherosclerosis, potentially increasing the risk of heart problems. This research suggests that B-cell depletion could have benefits beyond just fighting the flu itself.
So it's like a 2-for-1 benefit.
Potentially. It's definitely an exciting area for further research.
Okay, let's get back to those monoclonal antibody treatments for cancer. We talked about timing earlier, but there's another study that delves into this a bit deeper.
Oh, right, the one that looked at delaying treatment until day 8.
Yes, that one. What did it find?
Well, it seems like delaying treatment actually reduced how well the antibodies worked, even when they were used with other therapies.
So timing is crucial in multiple ways.
It is. This study suggests that the tumor microenvironment in the early stages of development might be key to the effectiveness of these antibodies.
So the earlier we intervene, the better.
Exactly. Once the tumor progresses, it becomes harder for those antibodies to do their job.
It makes you think about how important early detection is for cancer and how we need better tools for that.
Absolutely. Early detection is crucial. Okay, ready for a mind-blowing topic? Let's talk about DNA data storage.
Always. It's like science fiction becoming reality.
Right. There was this fascinating study in nature.com that proposes a new approach using coding theory and deep learning.
We talked about the advantages of DNA data storage before, how it's super dense and can last for thousands of years.
Right. It's nature's hard drive, way more efficient than anything we've built so far.
So how do these new techniques, coding theory and deep learning, help us with DNA data storage?
Well, coding theories is all about making sure the data can be encoded and decoded accurately. Even if there are errors, it's like error correction for DNA. And then deep learning comes in to optimize these processes.
So we're using these advanced computational tools to read and write data stored in DNA. That's incredible.
And this research is bringing us closer to making DNA data storage a practical reality. Just imagine, all our data stored in tiny vials of DNA.
It's mind-blowing. We're running out of time for this segment, but we'll be back with more exciting science news after the break.
And we're back with the final leg of our science news journey.
We've covered some amazing ground today, but there's still more to explore. Let's talk about the quest for a universal flu vaccine.
Ah, yes. I think everyone's hoping for that one.
A study in genomics, proteomics, and bioinformatics caught my eye. It's exploring a new approach that targets the CD20 protein on B cells.
CD20.
Yeah, it's a protein found on B cells.
Right, B cells, those are part of the immune system.
Exactly. And researchers are looking into whether depleting these B cells could actually help reduce the severity of flu infections.
Hold on, depleting immune cells to fight infection. Doesn't that seem, I don't know, A little backward. It might seem counterintuitive, but hear me out. Sometimes our immune response goes a little overboard. Like with the flu, you can get all this inflammation that actually causes more harm than good. So the idea is to fine-tune that immune response.
So we're not completely shutting down the immune system. We're just trying to make it, what, more efficient?
That's a good way to put it. The study found that depleting B cells using a specific monoclonal antibody treatment actually led to a reduction in atherosclerosis development in mice infected with the flu.
Atherosclerosis, and that's the buildup of plaque in arteries, right? I thought we were talking about the flu.
We are, but the flu can actually worsen atherosclerosis, potentially increasing the risk of heart problems. This research suggests that targeting B cells could have benefits beyond just fighting the flu itself.
Wow, a two-for-one solution.
Potentially. Okay, let's switch gears back to monoclonal antibody treatments for cancer. We talked about the importance of timing before.
Right, how early intervention seems to be a key.
Exactly, and this new study really drives that point home. Researchers found that delaying treatment with monoclonal antibodies until day 8 actually reduced how effective they were.
Even when combined with other therapies.
Even then, it seems like there's this critical window early on in tumor development where those antibodies work best. Once the tumor progresses, it becomes more challenging to target it effectively.
It's like trying to put out a small fire before it spreads into a massive inferno. Early action is crucial.
Absolutely. So this research underscores how important early detection is for cancer treatment.
And how crucial it is to develop even better diagnostic tools so we can catch those cancers as early as possible.
Couldn't agree more. All right, last but not least, let's revisit that mind-blowing topic. DNA data storage.
Always love to talk about that. It's like something straight out of science fiction.
It really is. A study in Nature.com presented a new approach using coding theory and deep learning.
We talked earlier about how incredibly dense DNA is. You can pack a ton of information into a tiny space.
And it's incredibly durable. We're talking about storing data for thousands of years.
Nature's hard drive. Way better than any technology we've come up with. But how do coding theory and deep learning fit into all of this?
Well, coding theory helps to ensure accurate data encoding and decoding. Even if there are errors during storage or retrieval, think of it as error correction for DNA. And deep learning comes in to optimize those processes.
So we're essentially building these really sophisticated algorithms to read and write data stored in DNA. It's mind-blowing.
It is, and this research brings us closer to a future where DNA data storage is not just a concept, but a practical reality.
Wow. Just imagine libraries and archives condensed into tiny vials of DNA, preserving knowledge for generations to come.
It's a future filled with amazing possibilities.
That's it for today's deep dive into the world of science. Thanks for joining us, and we'll catch you next time with more incredible breakthroughs and discoveries.
Keep exploring.